I'm gonna get going here. Friday. I'm sorry you couldn't hear the music today. You guys make too much noise, but you missed probably the most evil of evil men in all of the opera world, from Tosca. I wanted to say a few words before I start on membranes to sort of summarize all I went through on Friday, on Wednesday, in 2 minutes, no, 5 minutes maybe. Because I did go through a lot of material, and that material was sort of very different than things I've talked about in the first few lectures. So what I was trying to do on Wednesday was to tell you about a eukaryotic cell, the different compartments in the eukaryotic cell, and what their function is. That's one thing you are responsible for. You should know what all the functions of these various compartments in the cell are. And then I focused on this question of how are cells targeted to those specific compartments, with particular emphasis on post-translational and co-translational import. So those are the things you have to be clear about. You have to understand the difference between post-translational import and co-translational import, and which organelles receive proteins through one pathway versus the other pathway. So that was what all that was about. Today, I mentioned earlier that there is one lecture which deals with the structure and function of biological membranes, because membranes are clearly so important in cellular function, and that's what I'm going to be talking about today. I put on the board the topics that I'll try to cover. Music was Puccini, for those that don't care. The office hours today are 9 to 10. A lot of people are coming to office hours. And unfortunately, we can't get another room. The campus does not give us rooms for office hours. So we're just going to have to pack in there as well as we can, get to know your neighbor kind of thing. Let's start off with figure 56. 56 is a general figure that is useful because it shows you a cell, and it shows you the membranes that are in a cell, different kinds of membranes with different functions. And some of the functions of biological membranes are spelled out there. Obviously, the plasma membrane around the cell is a permeability barrier. It's a barrier. Some molecules can come in, and we'll discuss how they get in, and some molecules can't get in. There are also signal detecting components in the cell membrane, and I mentioned this originally when I pointed out that there are proteins which often have sugar molecules on them. These are in the plasma membrane, and the sugar molecules are part of this detecting device for saying this is a liver cell and that's a nerve cell. There are proteins in the outer membrane that are involved in transport, that allow things to come in and out of the cell. There are proteins in membranes. You heard about the ER membrane system on Wednesday that are involved in moving proteins into the ER, for example, so they can be glycosylated and targeted to various organelles. And there are specific functions in the cell associated with organelles, particularly the ones we will talk about, the mitochondria and the chloroplast, where there are internal membranes. And these internal membranes, as you'll see in a minute on this board, are packed with proteins. And they have a very high concentration of protein because they are involved in a very essential activity of the cell, which is to make ATP. ATP is the driving force for everything that goes on in biology. I put up here the phospholipid bilayer that I've talked about before. The phospholipid bilayer is considered to be the basic core structure of the biological membrane. However, when you look at, I'm going to put this up now, when you look at the composition of various membrane fractions, These numbers are not important. There's a point that I want to make from these numbers. You can look at various different kinds of membranes, like the plasma membrane from a blood cell, a liver cell, and you find roughly almost equivalent amounts of lipid and protein in those membranes. Phospholipid bilayer plus protein. Other membranes have more protein than phospholipid, the nuclear envelope, the ER. The most striking composition of a membrane fraction is the inner mitochondrial membrane. Remember, mitochondria have their outer membrane, and then they have this internal membrane system, which we'll talk about, where there's 3, almost 4 times more protein than lipid. So saying that biological membranes are phospholipid bilayers is not quite accurate. That's the point I'm trying to make. You would say the mitochondrial membrane is made of protein, and it's got a bit of lipid in it. So that's a very different situation than our simple thinking that you've got a phospholipid bilayer and then maybe there are a few proteins in that phospholipid bilayer. When people started to look at the structure of biological membranes, there was an early model of a membrane where the proteins were smeared on the surface of the membrane, on either side of the membrane. I'll make those into proteins. And this was an accepted model for the structure of biological membranes for a number of years. But as people worked with these membranes more and more, they realized that this was not an accurate representation of a biological membrane because there were proteins, yes, associated with the membrane, but there were 2 kinds of proteins that were associated with the membrane. There were proteins that were weakly bound to the membrane, and these are known as, there's 2 terms we use, and I use them interchangeably. Peripheral or extrinsic. So there were a group of proteins that could be removed relatively easily from membranes. That is, by washing membranes with high concentrations of salt, you removed a certain number of proteins from those membranes, which argued, yes, they were protein membranes, but they were weakly associated with the membrane. And then there was a group of proteins which were found to be tightly bound to the membranes. These are peripheral, integral, or intrinsic. These proteins are tightly associated with the membrane, very difficult to remove from the membrane. Thinking about the protein association of the membrane and the structure of the lipids that are in the membrane led Singer and Nicholson to propose a new model for membrane structure, which is shown in figure 58. It is called the fluid mosaic model of membrane structure, and this is important. This is what is now accepted as the most widely accepted model for a biological membrane. If you look at the top of that figure, you can see there are proteins which are actually inserted into the membrane and go through the membrane. I've drawn a protein like this, which actually goes through the membrane. There's a hydrophobic portion to it, and there are hydrophilic portions to it. This is the hydrophobic portion. This is a protein which is totally embedded into the membrane. The proteins that are in the membrane are associated with the membrane by hydrophobic interactions of the protein with the fatty acid side chains. The new concept that Singer and Nicholson came up with was that the protein is not static, not rigid, it's fluid. The best analogy I can give you to think about is you have icebergs that are floating around in an ocean. The ocean is basically the phospholipid bilayer, and the icebergs represent the protein molecules. So the lipids are certainly mobile. They can move in this direction or that direction. They can't flip from one side of the membrane to the other because that is an energetically unfavorable process. But there is a motion in the membrane in this plane by both the lipids and the proteins. The proteins can also move, but since the proteins are larger, they're going to move less rapidly than the lipids. So there is a fluidity to the membrane. That fluidity can be altered by various components, factors that I'll talk about in a minute. The other thing this was proposed originally, and I'll talk about this in more detail, was that when a protein spanned the membrane, a transmembrane protein, there was an alpha helical structure to that region. That's what's shown in the middle of that figure. It shows you've got a membrane. I'll just draw it like this. And there may be some of this stuff sticking out on one side, and then there's a helical region, and then there may be some stuff sticking out on the other side. This was a proposal, and I'll explain the basis of this proposal in a few minutes. So here we have an alpha helix, which exists, but it exists in the hydrophobic region of the protein, associated with the hydrophobic region of the phospholipid bilayer. Okay, what kind of factors affect the so-called fluidity of a membrane? Well, obviously, I think you could probably guess that if you have a high concentration of protein versus lipid, that membrane is going to be less fluid than a membrane that has a higher concentration of lipids. Probably more importantly, though, is remember when I talked about lipid structure, I talked about the fatty acid side chains. The fatty acid side chains being either saturated or unsaturated, and the figure 59 and 60 refer directly to that point, and I think 60 is probably the better figure because it shows you how fatty acid side chains can pack when there are saturated side chains. In the top of figure 60, you get a relatively close packing, and compare that with the figure below it, where you have unsaturated fatty acid side chains, where the spacing is basically pushed apart because of the kink in those fatty acid side chains. What this means is that a more fluid membrane, or a membrane will be more fluid with higher concentrations of unsaturated fatty acids. So those are two factors, that the protein concentration and the type of fatty acid side chain that is in the respective membrane. Another factor that can affect fluidity is temperature. It is possible to actually freeze out membranes. If you lower the temperature, you affect the mobility of the lipids in the membrane, and you can get to a point where there is no mobility because they cannot move anymore. So fluidity is another factor that can affect fluidity. One thing that comes out of the fluid mosaic model is that the membrane is asymmetric. And I have tried to show that. I haven't shown it very well in this. Yeah, well, I have shown it well in this figure. Well enough, let's put it that way. What you have on one side of the membrane, for example, is this particular hydrophilic protein. So this side is obviously different than this side. You have regions of these proteins, of the hydrophobic proteins, that go through the membrane. And generally, I should have made this look like something like this to show it's a different. You know, there's the part on this side. The regions on both sides of the membrane aren't the same because these hydrophilic regions that stick out of the membrane are different. This is different than that. It's got a different amino acid sequence here than there. In addition, you can have different lipids on different sides of the membrane. I haven't stressed this point that I've said phospholipids, but remember there are lots of different kinds of phospholipids. Go back to the figure I gave you on the structure of a phospholipid. I specifically talked about phosphatidylcholine, but there are 5 or 6 other phospholipids that are shown in that figure. Phosphatidylserine, phosphatidylethanolamine, and these lipids are not localized equally on both sides of the membrane. So there's an asymmetry to the sides of the membrane, and that's important in terms of some of the activities that we will be talking about later. Okay, I'm going to spend a fair amount of time talking about the structure of membrane proteins. If you look at figure 63, you can see there are 2 proteins that are shown in that figure going through a membrane. One of them comes through the membrane once. There's one helical region. But you can also have proteins where you have something here, and then you have a helix, and then you have a connecting, and then you have a helix, and then you have a connecting, and you have a helix, and then you have the other terminal end. You can have a protein that goes through the membrane 3, 5, 8 times. It doesn't, it depends on the structure of the protein. There are predicted structures of proteins shown in figure 64. One of them, glycophorin on the left, is a single pass protein, one transmembrane region, and it has carbohydrate side chains. It's obviously a protein in the plasma membrane, relatively simple. The other one is a more interesting, well, to me more interesting, proton. It's a protein known as bacteriorhodopsin, and if I have a few minutes when I talk about photosynthesis, I'll mention that protein again and what it does. Very unusual protein. That protein actually functions as a proton channel. It moves protons across the membrane, which is an interesting function. Proteins can also be organized in the plasma membrane to form channels. Sometimes one moves like a potassium ion. A channel is basically a hole in the membrane, but it's made of protein. And the specificity comes from the size of the hole and some of the interactions of the ion with various components in the protein itself. And you can see there are like 1, 2, 3, 4 or so alpha helices spanning the membrane, predicted to span the membrane to form a channel so a potassium ion can go through. Well, where does this idea come from that there are alpha helices that go across the hydrophobic region of a membrane? When people started to isolate membrane proteins, and I'll talk about that next, and determined the amino acid sequence of these proteins, what they discovered was there were regions of these proteins that were very hydrophobic. Very hydrophobic, meaning stretches of approximately 30 hydrophobic amino acids. And in order for a protein to span a membrane, if a region with 30 hydrophobic amino acids forms a helical structure, that is the distance across a biological membrane. So it has been suggested in those days that every time you saw a region of approximately 30 hydrophobic amino acids, you had a membrane spanning region, an alpha helical membrane spanning region. And when I say hydrophobic, these are no polar or charged amino acids. None. It's a bunch of AAs, all right. It's a bunch of valines, leucines, phenylalanines, things like that. The most hydrophobic amino acids are present in these regions. This is where things stood until people started to isolate and characterize membrane proteins more fully. It's important to realize that the isolation of membrane proteins is very difficult. Proteins that exist in the cytosol of the cell, they're in an aqueous solution. They're basically in water. It's not water, but they're in that something comparable to water. Easy to separate out from the cell, easy to work with because you're working in an aqueous environment. These proteins, these hydrophobic proteins, will have as many as 50 to 60% of their amino acids as hydrophobic amino acids. This is another characteristic of membrane proteins. If you just do an amino acid composition of these proteins, they have a very high concentration of hydrophobic amino acids. What this gets you at the end of the day is these proteins are not soluble in water. And firstly, it's very difficult to get them out of the membrane. The only way you can isolate these proteins is to disrupt this membrane structure. And then, if you disrupt it by various techniques, often these proteins will precipitate out of solution because they're not soluble in water. So what people developed are a bunch of compounds which are known as detergents. Everybody knows what detergents are, right? Well, these are the sort of same things you use to wash your clothes or your dishes. And what detergents are in biological systems is they are compounds which substitute for the phospholipid molecules that these proteins associate with in the biological membrane. So if you look at figure 68, you can see an intact membrane, and then you add a detergent, and the detergent dissolves away the lipids and binds to the proteins, and the detergents are lipid-like in the sense that they have a hydrophobic region and a hydrophilic region. So they will keep that solution as a detergent protein complex in solution. The kinds of compounds that people use, and there are hundreds of these. I spent a lot of time working with detergents, and we just had a shelf in the lab with different detergents doing basically the same thing, but sometimes they work in one system and not the other. And 3 detergents are shown in figure 69. The most interesting one and most widely used one is this compound sodium dodecyl sulfate, where you've got a long hydrocarbon chain, but it's attached to a sulfate group. The sulfate group is like the polar head group of the lipid, and the long chain hydrocarbon is like the fatty acid side chain. Sodium dodecyl sulfate is very commonly used in biological systems for solubilizing membranes. Now, detergents are very tricky to use. Sometimes you can get these proteins out of the membrane, their respective membrane, but they're not functional anymore because the detergent has disrupted the structure of this. But people, you have to do what you have to do in life. And you have to treat membranes with detergents to solubilize these proteins. There are several things you can do when you obtain an I'm going to say a sort of aqueous solution of membrane proteins to try to characterize these proteins. One of the most common ones is this procedure that if you go into any lab that's doing any kind of protein chemistry, protein biochemistry, you probably will do. It's called SDS page. And what that is it say what it is in the figure so I don't have to write it? No. SDS is sodium dodecyl sulfate. Page is polyacrylamide gel electrophoresis. What you do is shown in figure 70. You've got membrane fragments. You add SDS, so they are now solubilized with the detergent. And you have a gel. A gel is a medium that is, I don't even know how to describe it. It's sort of like Jell-O almost, really. And that's contained in 2 plates. Between 2 plates, you can attach electrodes to the system, positive, negative. You can put your proteins onto this gel system, turn the power on, positive, negative. Proteins will migrate through the gel depending on their molecular mass or weight. Big proteins move slowly. Small proteins move faster. So that's what's shown in figures 70. It shows you put the stuff on, you turn the power on, and these proteins start to separate on the basis of their molecular weight, which is a good thing if you want to know what's in a certain fraction. You take your gel out, you put it in something, a compound, that reacts with proteins. It's a blue dye. Most people use a blue dye. And wherever there is a protein, you get, depending on its molecular weight, you get a blue band. And what's shown in the right are the number of bands you see. This is a fraction membrane proteins from human erythrocytes, red blood cells. So you can see there are a lot of bands on this. Each one represents a different protein. This is a real, come on, come on, come on. There it is. This is a gel from my former life as a research scientist at UC Berkeley, okay? So I started off with chloroplast fragments, membranes from chloroplasts, and I have done, what you can do is you can dry the gel at the end of the day and keep it. This is 15 years old. I'm going to have to use my finger. Each one of these represents a distinct protein in the fraction that I have. These are just molecular weight standards, okay? So you now at least have the possibility of saying, well, there are all these different proteins in this fraction. What are they? Two questions come up. What are the proteins and what's their function? So if you're looking at an erythrocyte membrane, you might think some of these things have to do with transport across the membrane or something like that. So you can start to relate specific proteins with their function. How do you do that? Well, it's possible before you stain this with blue stuff, Kumasi blue it's called. You can cut out these little bands. You get a little razor blade and you can cut them out and you mash them up and you can actually get some and you feed them into an amino acid sequence machine. You can get some amino acid sequence data. Not a lot of it. It won't give you the sequence of a total protein. But you may get enough amino acid sequence data to compare the data you get for this specific protein and the enormous database that's available on the sequence of proteins. And that way you can say this protein is spectrin or actin or something like that. And it depends on how many amino acids you can get off of a tiny bit of a gel slice. But if you're lucky, you might get 5, 6, 7, 8, and it might give you a unique sequence that you can identify from the database. Okay. Once you start to do this, you can start to relate the function to the activity. If you're working with a fraction that has a defined activity, like a plasma membrane, and you're looking for membrane transport proteins, you may be lucky and you may be able to obtain fractions that are missing certain proteins. And when you miss certain proteins from this fraction, certain activities may disappear, and then that's how you can sort of start to get a handle on what all of these various proteins are doing. The most significant breakthrough in membrane biochemistry came in the mid-1980s when two German scientists isolated a membrane protein and did something that had never been done before. They crystallized this protein. Membrane proteins are very difficult to crystallize because, for one reason, they've got all this detergent stuck to them, and to get a good crystal, you need a homogeneous sample. And you may have different molecules with different amounts of detergent bound to them. So it's been very difficult to crystallize membrane proteins, but they worked out techniques for crystallizing a membrane protein. And the protein they picked was interesting to me because it was a protein in photosynthesis. May not be interesting to you, but it turns out to be a very interesting protein. It was a protein from a photosynthetic bacterium, a bacteria that does photosynthesis. And it was a protein you'll hear, in a general way, you'll hear more about this later, but let me just say it is the protein complex where light comes in as gets converted to chemical products. It is the so-called primary reactant of photosynthesis. So it's a very important process. And this was a structure that they determined. There are 4 proteins in this complex. There's a protein up here, there's a protein down here, and there are 2 proteins here. What's interesting about this structure is all of this corresponds to alpha helices. All of these different tubes that you see there in this figure are alpha helical regions which had been predicted from the amino acid sequence of these proteins, which existed before, because of these 30 amino acid residue hydrophobic regions. And this was the first time that they had been experimentally verified. So when people predicted that there were alpha helices, no one had ever seen an alpha helix. But once this structure was solved, you could see all of these proteins were alpha helical, and they presumably span, this would be the membrane, and they're spanning the membrane just as predicted for structures like going back to figure 64. The prediction of alpha helical regions in proteins was done by something, I have to mention this, it's slightly out of order, but it's okay, because it's done very often by something known as hydropathy profiles, fancy name. But what people are able to do using various computer programs is feed a sequence into these programs, and these programs will predict these alpha helical regions. And this is what is shown in figure 66, which I skipped, and now I'm talking about. But this is taking a protein of known sequence, doing a hydropathy analysis of it, and you find 4 regions, the regions that are shaded, that are very hydrophobic regions and are predicted to be membrane spanning regions. So there was a lot of information available based on indirect methods that these membrane proteins had membrane spanning regions, 4 of them in the protein that is given in figure 66, 1 of them on the protein on the previous page. But until an actual structure came out, it was hand waving. And there are models and there are models, but you have to test your model and you have to prove your model. And much of the biochemistry that came before the structure, I think, has turned out to be reliable and true, which is nice if you're a biochemist. So this was the first protein complex for which a structure was determined, and enormous amount of information came out of this structure. I mean, the protein biochemists were ecstatic because they had a structure and they could look at how transmembrane spans form. They could look at how things are not in the membrane, the kind of interactions. The people who are more interested in function began to understand how this complex functioned as a energy converting unit. There's a picture in their original paper where they show, I just remember, it's the polar side chains of all the amino acids in this complex. And as I said, there aren't many. And what you see is there's, let's do this. So you've got this, and then you've got all this stuff in the membrane, and there's stuff outside the membrane. You can see the stuff outside the membrane. So you see a bunch of charges here, and you see a bunch of charges here. And the region of this complex that is in the membrane, it's black. It's totally black. No charged amino acids in the hydrophobic region. No charged or polar, I'll say no polar amino in the membrane region. And this was something that had been predicted, but again, the structure proved it. It proved that you simply do not put a polar or charged amino acid into this membrane region. And then, of course, I'll come back later and say, you know, there are cases where there are charged amino acids. If there's a charged amino acid in this region, it's doing something very, very important. That's all I can say at this stage of the game. So this work was done in the early 1980s. A paper came out in 1986. It was reported at a meeting where it was first reported, and it was just spectacular. I mean, it was spectacular. Everybody went, I don't believe it, you know. You can't do this. You cannot crystallize and get the structure of a membrane protein, but here it was. And it took the Nobel Prize Foundation 2 years to give these scientists a Nobel Prize in chemistry. They never do that. They wait, you know, 50 years to make sure that the results are stable. And no, this was very important. So Deisenhofer and McHale are their names, and they not only crystallized this particular complex, but they worked out a lot of the procedures that people use to try and crystallize proteins. The number of proteins for which we have three-dimensional structures is very high. Probably 90% of those proteins are soluble proteins. And, you know, maybe 10% are membrane proteins, but probably no more than 10%. Okay, so that's kind of a nice story. I like that story, obviously. One of the problems, and I'll probably not get through this today, but that's okay. One of the problems that we have to deal with, though, with membranes is, I've said, is getting molecules across membranes. The cell membrane, the plasma membrane, is not a barrier. It's a semi-permeable barrier because molecules have to cross membranes. And there are several ways that one can get things across a membrane. For example, so we're talking about membrane transport now. Now that you understand all there is to understand about the structure, the easiest, simplest way is, one, simple diffusion. That is, if you have a high concentration of a molecule on one side of a membrane, and you have a low concentration of a molecule on the other side of the membrane, the thermodynamic driving force wants to equalize those concentrations, and you will get molecules moving across the membrane. Simple diffusion is entirely dependent on concentration difference. So you go from high to low. It's simple. It will also depend on the hydrophobicity of the molecule. So, hydrophobic compounds, such as some of those shown in Figure 71, large, uncharged, no. Hydrophobic molecules in general will cross membranes easily. Charged molecules, the larger they are, like sugars, will not cross membranes to any significant extent. Ions, which are very important in all biological cells, cannot cross membranes hardly at all, let's say. You know, I'm saying all or none. It's really not all or none, but it's simpler to say all or none. Exceptions are always interesting, but we're going to say all or none. There is another process which is known as facilitated. It's either called facilitated transport, or it can be called, but I think to contrast it with this, facilitated diffusion. Facilitated diffusion what does this mean? What's facilitating it? Protein. It's a protein-mediated process. That means there are proteins in the membrane that are making the transport of a molecule across that membrane possible. The proteins can occur in channels, or they can occur as a carrier. And you have already a figure that showed you the potassium channel, where there are proteins which form a channel, a pore, basically in the membrane, and the potassium ion can go through it. That's the channel. The carrier, I'll talk about these more next time, is where the molecule moving across the membrane interacts with the protein. And essentially, there are changes in shape of the protein in the membrane that allow the molecule to cross the membrane. Facilitated diffusion occurs in the same direction as the concentration gradient. So a facilitated diffusion is dependent on a concentration gradient, and that means you can move molecules from high to low only. So in that sense, it's a diffusion. It is a diffusion process. Diffusion is high to low without a protein. Facilitated diffusion is high to low, but proteins are involved. The third type of transport process, which is very important in cells, is something known as active transport. Active transport is protein mediated, but it can work against the concentration gradient. Can work against, instead of with, against a concentration gradient. That is, if I have a high concentration of an ion in the cell, by the process of active transport, I will transport more of those ions into the cell. That's not thermodynamically favorable. You're working against the concentration gradient, and for that reason, this process requires energy. It takes energy to move a molecule against its gradient. Generally, we have not talked about ATP, but generally the source of energy for these processes is this compound ATP, adenosine triphosphate, which we will talk about in more detail. The last thing I'll mention, because it's a good place to stop, and then I'll finish this on Monday, is when you look at how molecules are transported, if you look at simple diffusion, so this is the rate of diffusion versus the concentration. Simple diffusion is like this, just diffusion. Wow. Did I call it simple? Yeah, I call it simple diffusion. So the higher the concentration difference across the membrane, the higher the rate. When you look at protein-mediated processes, you do not get this relationship. You get a relationship that looks like this. The rate is faster initially, and then it levels off. The term that's used for this, so this is facilitated or active transport. What happens here is something that's known as saturation. term that you will see again when we talk about enzymes next week. That is, the rate goes up, and then it doesn't stop. Don't get this confused. When a system becomes saturated, it's continuing, but it's occurring at the maximum rate it can occur. Why is this? The number of protein molecules in a membrane are limited. You don't have an infinite supply. Down here, they're working very efficiently, and there are plenty of them. When you get up here, even though you increase the concentration of the molecule you're trying to transport, all of the proteins are occupied transporting things. So the rate cannot be increased above a certain level. And this is true. This is also true. This behavior we will see again when we talk about enzymes and enzyme regulation. Enzymes, their activity is saturated at certain concentrations of substrate. Transport proteins, their activity is saturated at certain concentrations of transport proteins because they simply don't have an infinite supply. But they work better at low concentrations because they're designed to interact with a specific molecule. There's a specificity that comes about. For example, even the channel, the potassium channel will work with potassium, but it doesn't work with sodium. And that's got to do with that's what the protein brings to the game. Okay. All right. So next time I'm going to stop here. You've got 5 minutes to enjoy yourself. I'll talk a bit more about transport, but it won't take me very long to finish it.